modified version. Why? For better understanding. डिजाइन जो पहले किया उसको मॉडिफाई किया, और व्हाई, क्यों? बिकॉज वी वांट इम्प्रूवमेंट इन आवर नॉलेज, इन आवर मेथड्स, टू सिंप्लीफाई द थिंग्स, टू गेट रिजल्ट as quick as possible, as simple as possible. Designed to simplify removal of excess primer and wild type template after each PCR step. Step is denaturing, annealing, and elongation. So in these steps, what we do, we remove if we have extra primer or wild type of the template, because we need only mutant here. And for this removal, we use these two type of things. One is primer, one is bead. Primer is biotinylated primers, and beads are magnetic bead which are coated with streptavidin. Improve the speed and efficiency. This is how we can improve. What efficiency will increase in this way, and speed will also. We don't have to waste our time on the non-desirable things, which are excess primer and wild type of DNA sequence. So, overextension mutagenesis. So, overlap extension only will be used to improve speed and efficiency. Now, there is some more things about the overlapping extension method to understand more better. What is this? A PCR-based technique used to introduce desired mutation into DNA. We know this. Primer use. Outermost primer is called flanking primer, like this part and this part. So this one is overlapping part, this one is DNA part, and this one is primer part. So flanking one is this one. So one is complementary to the antisense strand. Okay, this is complementary to the antisense strand which will synthesize. And 4, this one is 2, this one consider 3, and this one consider 4. 4 is complementary to the sense strand. Whatever is, if this is considered sense, then it will consider a antisense. So this is just nothing. And 2 internal primer. These are the internal thing. These are the internal primer. 1 contain desired mutation, in which it contains desired mutation designed to introduce mutation into each strand. Both strand will get mutant by what? By repeatedly PCR मैकेनिज्म जैसे जैसे चलेगा, पहले स्ट्रैंड में ये म्यूटेंट वाला रेप्लिकेट एज ए स्ट्रैंड यूज हुआ, then next time this will consider new one which synthesize here will be used as a template in next PCR. So this is how mutate one will selected and wild type will be removed from this PCR sequences. What is the procedure? This is the main thing. First, what we do, 2 separate PCR reaction will carried out in 2 tube A and B. There is 2 tube in which 2 separate PCR will be run. Both reaction use same reaction condition. There will be same reaction condition like temperature, pH, everything will be same in both tube. Each tube contain 1 flanking primer. In this 1 flanking primer, is here and one internal. This one is internal and this one is flanking one, mutating primer, and two different PCR fragments are produced, and this is in A and the same condition of the primer is in B also. Okay, then in second step we will mix and re-anneal. PCR product from both tube will be mixed. From these two both tube we mix the whatever there is content will mix in next step and re-anneal. Then we denature and separate all these and then re-anneal them. Stand from first reaction anneal with stand from second reaction. So this stand from the A and this stand one from the B will anneal with each other, and downward was 2 from A and 2 from B will be anneal with each other at overlapping region. Then extension. How we do extension? The hybrid molecule contain 5' short and 8' primers. So these are the short primers and these are the long. So this one is 3' end. So it will make this strand with the help of tag polymerase, and this is 3' will extend from this to this way. Both will be formed. So here there is a formation of two primers. Sorry, two strands. In presence of both strand mutation, now mutation is present in both strands, in this one and in this one both, but there is one strand is mutant, there is also one strand. Both strand will mutate, but both what? These are both considered, this one and this one, not this one or this one. Okay. Final PCR. A final PCR performed using flanking primer, primer 1 and primer 4. The full length production is amplified and mutant DNA is obtained. And this is where the next is, in which next step we remove this part, because this will, in next step this will be considered a work as a template, and this one separately work as a template. So this one will make what? Mutant one, but this one make wild one. So that wild one will be separate out, and then after at a final stage when we get so many PCR reaction are run, so after that rain. Okay, after that we only have mutant DNA. Okay, now what is mega primer PCR? We will see this topic in next class. Okay, bye bye, take care.